All right, let's get this meeting started. Welcome to STAT's virtual event. We're calling this the ESMO Recap Live, sponsored by Natera. Thanks for joining us. I'm Adam Feuerstein, Senior Writer at Biotech here at STAT. Just a few minutes, I'm going to have a couple of two experts to help us discuss some of the research that was presented at the recently completed ESMO cancer meeting. A few quick housekeeping items for you before we get started. At the bottom of your screen, you will see a few icons. There is a chat icon and a Q&A icon. The chat feature you can use amongst yourselves if you want to chat. But if you want to ask questions of our guests today, and I really encourage you to ask questions, because I will use the questions. We usually have a really great number of questions, and we'll use those throughout the event today. So post your questions in the Q&A. Hit the Q&A button, type your question in. I will see them on my screen, and I'll be able to use them and ask our experts, our guests today, ask those questions for you. And again, we'll be taking those questions throughout today's event. Again, thanks all for being here. Before we get started, I want to welcome Dr. Alex Aleshin. He is Medical Director of Oncology at Natera, and he's going to be giving you a brief introduction. Alex, take it away.
Thank you, Adam. Hello, everyone. My name is Dr. Alexi Aleshin. I'm the Senior Medical Director for Oncology here at Natera. We're extremely excited to partner with STAT News today for this discussion with really just some of the significant data presented at ESMO this year. This year's conference has really been particularly notable given the breadth of novel clinical trial results and technologies that were presented upon. One such technology has been featured very prominently in this year's meeting is circulating tumor DNA. And this technology has really fundamentally changed how we think about both clinical trial enrollment as well as early assessment of treatment efficacy. So one study where I think a lot of observers are very interested in ongoing CTDNA mutational as well as dynamic assessment is the CodeBreak 100 study, which will be discussed in further detail later in this webinar. But really beyond this study, CTDNA has been featured prominently at this year's meeting with over 50 abstracts and talks being dedicated in part or exclusively to the analysis of CTDNA in all phases of drug development. Next slide, please. Great. How do you think about possible applications of ctDNA in clinical practice as well as pharma drug development? Well, I really like to think about this as a spectrum. So on one side of the spectrum here, you can see on the left, the ctDNA is being studied for the asymptomatic detection of cancer before histological diagnosis is made. And this is really to augment screening technologies such as mammography and colonoscopy. On the other side of the spectrum, we think of very kind of common applications of ctDNA. And this is using ctDNA to identify actionable mutations, for example, EGFR mutations in lung cancer, for therapy selection purposes, typically in patients with advanced or metastatic diseases. And obviously, this application of ctDNA over the last few years has revolutionized our ability to identify patients with actionable mutations, as well as study tumor heterogeneity, especially when tissue is limited or unavailable. But really in the middle of this spectrum, for the first time in the past, I would say really 1 or 2 years, the technology for ctDNA assessment has really advanced to the point of now allowing ctDNA assays to be adopted for 3 novel use cases that really stand to revolutionize how cancer is not just treated, but really how clinical trials are conducted, especially in the early stage disease setting. And really these use cases are listed here include the detection for the first time of minimal residual disease in solid tumors. And really until recently, this thought to be almost an impossible goal to accomplish with ctDNA. And really was only thought to be relevant to mostly hematologic malignancies. For the first time, this is something that's possible and being done both in clinical practice as well as in pharmaceutical drug development. Also, the second use case is using ctDNA to identify molecular recurrence. And this is identification of ctDNA before clinical radiological relapse, sometimes months, if not years earlier. So really this provides a window of opportunity where a patient may be molecularly relapsing, but still has not clinically relapsed. And then lastly, using really these precise ctDNA assays for the first time, it's now possible to monitor treatment response, really augment typical RECIST criteria, and really understand earlier in drug development if the treatment is efficacious or not. Next slide, please. So really the big technological advance in this field that has opened up these novel applications is the development of ultrasensitive and specific ctDNA assays that really improve on currently available tumor, as we call, naive ctDNA approaches. One such approach is really a tumor-informed ctDNA method, which is being developed by Natera and known as Signatera, which is now being commercialized and developed by a company for multiple use cases listed previously. The way that this approach works is really by identifying a genomic fingerprint of a patient's tumor, really identify all the personalized somatic mutations that are particular to that patient's tumor. Once those mutations are identified, this allows for a personalized or bespoke ctDNA assay to be developed to really target and track those clonal mutations over time. This really allows for an ultra-high sensitivity approach by leveraging this a priori knowledge of what mutations to look for in a patient's plasma ahead of time. It really allows for this multiple shots on goal to improve detection of ctDNA down to a single molecule level. This also really maximizes the specificity of this approach by eliminating common sources of false positives that really plague many non-tumor informed approaches, including CHIP mutations or clonal hematopoiesis of an indeterminate potential, which without this type of filtering are really indistinguishable from many clonal tumor-derived mutations and are a frequent source of false positives with these other assay approaches. And furthermore, this also allows for highly accurate CTA quantification by reducing noise in ctDNA measurements, which really stems from the ability to multiplex multiple variants in parallel. And really, for the first time, this allows for treatment response monitoring, even in the adjuvant setting. So when the patient does not have a radiological correlative disease that can be measured, ultrasensitive ctDNA approaches can be used to really understand how ctDNA is changing in response to therapies in the adjuvant setting. Next slide, please. However, really in the end, prospective clinical validation of such technologies is extremely important to really establish the performance characteristics of any new assay. And I think this is a great slide that really shows KNM curves for patients with early stage disease derived with a Signatera technology based on MRD or minimal residual disease assessment in these variety of solid tumors, which really shows that, I think the following key central tenets of MRD analysis in solid tumors. Really, the first one is that in any patient that is ctDNA positive after definitive treatment, that can be surgery, or for example, surgery and adjuvant therapy in patients with early stage disease, the risk of recurrence in these ctDNA positive patients is close to, if not 100%, as you can see here. I think seeing KNM curves like this is extremely rare. You can almost drive a truck through them, which really shows kind of the differential prognosis of patients who are ctDNA positive versus ctDNA negative. Furthermore, ctDNA detection precedes clinical recurrence by months, if not years. We think of this as the lead time, which really provides a window of opportunity for early treatment intervention in patients who are otherwise destined to recur and develop metastatic disease. And really, this data has not gone unnoticed. I mean, the concept of MRD positivity has really now been considered as almost a new indication for the typical kind of first-line, second-line metastatic disease settings that we think about when we think about new drugs being developed. And this ability to really test drugs and new agents and new approaches in these highly enriched patient populations has really fundamentally changed how adjuvant drug development is being thought about, as well as how certain studies are now being designed. Really, for the first time, adjuvant studies can be conducted with just 50 to maybe 100 MRD positive patients. So you don't need the typical 2,000 to 5,000 patients with years of follow-up to show that a new approach works in the adjuvant setting if you limit a particular drug development to these MRD positive patients, because again, the chance of recurrence is so high. And adjuvant studies using this approach are non-dilutive of patients who are otherwise cured and do not stand to benefit from new adjuvant drugs being studied. Next slide. So really, I think, kind excited to advance this field forward and really work with our biopharma partners to really, for the first time, personalize adjuvant as well as extended adjuvant treatment regimens for the millions of patients with early-stage cancer diagnoses. Really, technology like ours has really started to change the landscape of how these early-stage clinical trials are being conducted. As you can see here, this is just a small list of the studies that ctDNA MRD technology is being incorporated into, really in order to look at kind of four things. The first, can we identify patients who are really at high risk of recurrence and really only test novel therapeutic approaches in these patients? And we really think about this as MRD for enrollment. The second use case is really using ctDNA dynamics as an early biomarker of response. So instead of waiting for DFS or OS events to occur, we look at patients who, for example, clear their ctDNA signal to really understand if a new therapy is working or not. And then lastly, can we combine genomic profiling information with ctDNA MRD assessment to identify, for example, novel therapeutic approaches in, for example, KRAS mutated or EGFR mutated patients who are also ctDNA positive. And then lastly, can we leverage technologies like this to look at additional plasma-derived biomarkers, for example, tracking putative neoantigen mutations in patients who maybe receive, for example, tumor-specific vaccines. So really, I think in conclusion, ctDNA MRD detection and molecular surveillance as well as treatment response monitoring really tend to fundamentally change how early clinical trials are being designed and conducted. The concept of molecular recurrence is rapidly gaining attention and is now being viewed not just as a novel kind of frontier, but really, I think, something that's here today to really change how adjuvant studies are being conducted and bring personalized novel therapies to the millions of patients with early stage cancer where drug development typically lags behind that of the metastatic setting. So with this, I really want to thank everyone for your attention today and want to turn it over to Adam to lead the rest of the discussion. Thank you.
Dr. Lushan, thanks so much for your time. We appreciate that. And so now I'd like to introduce the speakers for the rest of our panel. I'm going to let them introduce themselves. We have Dr. David Hong and Dr. Mary Jennifer Markham, and we'll bring them on and they can introduce themselves. MJ, why don't you start?
Sure, thanks Adam. Thanks for having me. I'm Mary Jennifer Markham. I'm a medical oncologist at the University of Florida in Gainesville, Florida. I serve as the interim chief of the Division of Hematology Oncology, and I'm the Associate Director for Medical Affairs for the UF Health Cancer Center.
David?
Yeah, I'm Dave Hong. I'm also a medical oncologist who primarily focuses on early drug development here at MD Anderson Cancer Center. I'm the Deputy Director of the Department of Investigational Cancer Therapeutics and the Associate Vice President of Clinical Research here at MD Anderson.
Great. And like I said, as we go through this presentation, discussion, any questions you guys have, throw them in the Q&A and we'll try to get to them. So I don't know about you guys, I thought ESMO was actually, I kind of had low expectations for ESMO this year, but there was actually a lot of interesting data presented. I had to work over the weekend, which I don't like, but it was fine. But I wanted to maybe get started, David, with you. I've been writing about KRAS inhibitors, the development of KRAS inhibitors since early last year. I think it's probably, it seems to be one of the more exciting areas in targeted drug development, particularly lung cancer. A lot of excitement, a lot of it's being closely followed. The programs that are going through, and so you presented the latest data from Amgen's AMG 510 drug, their final phase 1 data at ESMO this year, and I wanted to just to kind of go through those data and discuss those data with you as part of this event. So maybe let's start with some background on kind of what we're talking about here. This KRAS mutation, the KRAS G12C specifically, that this drug targets. It's called the undruggable protein. And so maybe tell us a little bit of background there.
Well, people have known about KRAS for, 4 decades. And, if you take the whole number of different codons and alleles that are involved in KRAS, that's, close to 40% of all tumor types that have existing, at least metastatic cancer patients that have existing KRAS. And so I think I think everybody has been trying to understand it for a very long time and trying to particularly target it. I think it's been very difficult to target because it's this GTP pocket that exists in the KRAS protein. For whatever reason, I'm not a medicinal chemist, but I think it's been very, very difficult to develop molecules that can kind of fit into that pocket and put that KRAS in an inactive state. As you recall, Adam, KRAS exists in this kind of inactive and active state, and when it's bound to GDP, it's inactive. When it's phosphorylated GTP, it's active, and then triggers a cascade of other molecules downstream that primarily signals through MAP kinase. And for whatever reason, that GTP pocket has been very, very difficult to target. Dr. Showkat, who's at the University of California, San Francisco, several years ago, I would argue is probably the landmark paper, not necessarily, and not to say that dismiss the clinical trial, but that really showed that this pocket could be targeted. So he was the one who created the first renditions of the molecules that could fit into this P2, SHP2 region of the pocket, and was able to identify molecules could do that. So it's just taken that long, and you never know if something's going to work in patients until you actually get it into patients. And we were able to get the first molecule that has been really in the clinic now for almost about a year and a half, 2 years, AMG 510 or sotorasib, into the clinic, and I was able to had the honor of presenting that data at ESMO this year.
Right. So, I mean, one of the things, it is so closely followed because sotorasib is the first drug of its type to enter into clinical trials. So people have been following it. There are other companies developing their own. So maybe just like summarize the data that that you presented over the weekend.
Yeah. So the data was really kind of a summary of the non-small cell lung data, more so than the whole data set, which was published in New England Journal. It was kind of focusing on the phase 1 population in non-small cell lung cancer. The trial was, a typical phase 1. We had 4 doses in dose escalation at a Bayesian design, and we enrolled about 59 non-small cell lung cancer patients. The vast majority of these patients were very heavily pretreated. 100% of them were platinum refractory. Close to 90% of those patients had some kind of immunotherapy. All of them had KRAS G12C, obviously, and the median number of prior therapies was around 3, greater than 3. So what did we see? So safety-wise, the overall trial showed really excellent safety. I do a lot of phase 1 studies, and I do see a number of molecules with lots of toxicities. We weren't able to reach we didn't find any DLTs. We reached the highest dose, which was 960 milligrams once daily. The vast majority of adverse events that all treatment emergent adverse events were still grade 1 and 2. And there were a subset of population of patients, about 18%, who did have all emergent adverse events of about 18%. The side effects were primarily things that you typically see with oral medications, some diarrhea. We did see some elevation in the liver function tests. But probably the important point of the adverse event profile is that overall the drug was very tolerable, and it's even patients have been able to really tolerate this in many of the patients for long periods of time. So the response rate in the non-small cell lung cancer cohort was overall was about 32%. At the 960, the confirmed response rate was 35.3%. And the median duration of response was, I think, 10.9 months. And the overall progression-free survival, because we didn't have that data earlier at World Lung, was now about 6.3 months.
So Dave, let me ask you a question about the, the durability, because I think coming in to your presentation, durability of response on this drug was kind of the concern or the question. People were wondering what kind of durability we were going to see from the data that you presented. So put the durability that you mentioned, put that into context for us in terms of like how strong that is.
So let me step back and say, one, I'm not a lung cancer expert, but I've had lots of discussions with lung cancer experts. And, one of the co-authors and the co-PI of this trial at MSKC, Bob Lee, is a lung cancer expert. And all of them have shared with me that they believe that this, both the response rate, close to 35% now, and the 960, which is the phase 2, and the median progression-free survival, 6.3, is a significant, meaningful durability and response in a refractory patient population such as this, right? And so it's not probably the drug that has had the highest response rate I've ever had the pleasure of working with is larotrectinib, right? It's not a 90% response rate drug, obviously, but at the same time, this clearly, at least from all the lung cancer experts I've spoken to, feel that this is a meaningful, significantly meaningful duration of response in progression-free survival and also response rate.
So when I spoke to some lung cancer experts for the story that I wrote over the weekend, some of them expressed a little bit of disappointment in that, and not. And sort of preface what disappointment means, but maybe relative to expectations. And you mentioned larotrectinib, which is another targeted lung cancer drug. And we see in these highly targeted lung cancer drugs, we see really, really high response rates. And here we're seeing response rates that are, call it a third, 30%.
I think we're all getting spoiled.
Yeah, I mean, well, maybe just comment on that and what do you think that means?
With these new drugs that are having these great response rates, my colleague Dr. Sabaya, recently published also on RET fusions, and I'll tell you, I think two things. One is that fusions like larotrectinib, RET fusions, Alkros, whatever. For whatever reason, that I think, and several investigators have published on this. Rizal Kurdrak was my previous boss. A number of people, they uniquely are just huge drivers of the underlying disease. I want to say that I still think that we will see higher activity, and this is my best guess, in patients who are less pre-treated up front with sotorasib than we see in this population. And so I think that you're going to see, like with other kind of targeted agents such as EGFR, et cetera, as you go up that kind of line of second line, third line, fourth line, third line, second line, first line, you're going to see probably higher response rates, maybe even longer progression-free survival rates. But I just think that we've been, these RET fusions have really kind of knocked us out because they seem to be such high responses. Remember, the MET exon 14 deleted patients, I think tebotinib was still around in the frontline setting, it was only like 46% response rate, published in New England Journal. So I wouldn't, I think there was high hopes because everybody was hoping that this would be like, super hit out of the ballpark and into from Houston to Boston or something, but it's still, I think, a home run in this case. So we'll see how this plays out as this drug goes up the chain.
MJ, do you have any thoughts? I want to kind of, I don't want to leave you out of this discussion and bring you in. Any thoughts about kind of the relativity, when you see response rate like this and what this means?
I think that honestly, anything that gives our patients a bit of hope is something that we'd like to have on the table of options available. And I think David's point about, this is early, we need to see how it plays out is a really important one. But honestly, the more options for these relapse and refractory patients we have available, even if response rates are lower, but if there is meaningful activity, it can mean a lot to an individual patient.
Mm-hmm. David, I want to ask you, one of the unfortunate features of all targeted drugs is that patients relapse. And I want to ask you if you could offer any details about the kind of relapse that you're seeing. You know, patients have relapsed from this phase 1 study. The kinds of relapse that we're seeing are patients, are they mutating away from the G12C mutation to some other KRAS mutation, or has that been determined yet?
Yeah, that's a very good question. No, we're exploring that. You know, in the ongoing phase 1 and in combinations. Obviously, the combinations may also give us a clue, but we're ongoing, you know, trying to determine that either through progression biopsies, and also we are following them with the cfDNA ongoing in these patients. I can't, you know, really kind of share with you at this point, since we haven't published any of that data, what we're seeing per se. It is also, I want to caveat this because MJ also sees these patients, refractory patients, I'm sure. It is really hard to try to convince a patient, unless they're able to transition to another trial right away, to get a progression biopsy. You know, they're either, you know, most of these patients that I see at least are kind of, this is their last train before they go to hospice, right? But we are doing our best to try to get progression biopsies, cfDNA, and other correlatives that will help us determine what is kind of the next step. And I think, you know, kudos to companies both like Mirati and Amgen is that we are rapidly moving into combination trials. And some of those patients were able to transition to combination trials, and we'll see if likely there will be any benefit in that context, and gives us clues to mechanisms of resistance.
I want to ask you about a question about dosing. You know, the highest dose tested is 960. I think, you know, your paper was published in NEJM over the weekend too. There was an editorial where I think there was a question about dosing, and what, you know, the authors of the editorial questioned whether or not this drug was being underdosed. And I wondered if you could maybe comment on that, your thoughts on whether dosing could go higher. I think Amgen is, this is a, correct me, I think this is a once a day drug. Amgen has maybe talked about twice a day dosing. Can you respond or comment on anything related to that?
We are, you know, as all phase 1 trials, you know, for example, we are looking at PK relative to, you know, like food effect studies, et cetera. I'm not sure if I can comment on further dosing. I don't know if that's been reported yet, but you know, these are all kind of good questions. Theoretically, if you look at what we've seen so far preclinically, you know, phospho total inhibition occurs at around 360 milligrams, right? So anything above that, theoretically, we're still, you know, inhibiting or putting KRAS into that inactive state. Now, we did see, if you look at that waterfall graph, we did see responses at 360. And so it is that play between, you know, how much of this is dose versus, you know, what the drug is doing. It's an inactive covalently bonding, irreversible covalently bonding inhibitor. So it doesn't make any sense for us to kind of crank this dose continually up to the point where we really see significant DLTs and MTDs. You know, I think the decision is that, you know, we need to move on this. The response rates look pretty good. The trial was designed to the maximum of 960. And so I think that was the decision made by Amgen to move forward this as fast, as expeditiously as they could.
Okay. And I'm going to come back to you, David, and we're going to talk a little bit about kind of what the future might hold for this drug. But I wanted to bring MJ in, and I'm calling you MJ because that's what you told me to call you. So
yes, I did.
So thank you for that. But I wanted to kind of maybe just, again, this theme of what was presented at ESMO this year. And I always like conferences where there's drugs that are sort of targeting the same patient populations, and you can kind of, obviously it's not apples to apples comparisons, but you can compare drugs. And this was something that happened over the weekend or at ESMO this year in cervical cancer, where we have a number of drugs that are being developed for patients in cervical cancer. So maybe just give us a little bit of a synopsis about cervical cancer, just to start off as background, MJ.
Sure. So the challenge with cervical cancer is that once it's advanced and once it's recurrent, it's a deadly disease. The standard treatment right now for most women with cervical cancer is if it's early enough, then surgery plays a role. Once the stage is a little more advanced, some combination of radiation with chemotherapy given along with it, usually cisplatin, is primary. A large number of women, though, with advanced disease will have recurrence. And once a patient has recurrence, or if they present with more advanced disease that is not curable, the standard really is traditional chemotherapy. And the most common agents used are cisplatin or carboplatin with a taxane. Bevacizumab did change the landscape when there was a survival advantage showed to adding bevacizumab to chemotherapy. And then I guess most recently was the introduction of pembrolizumab for those women who have PD-L1 positive tumors. The challenge is not all patients can receive bevacizumab due to coexisting medical problems or non-healing wounds, things like that. And certainly not all women can receive pembrolizumab. All tumors are PD-L1 positive. So there are several unmet needs within the field.
And I think there was a paper also presented at ESMO, which kind of looked at cervical patients and looked at PD-L1 response. So again, so maybe you can kind of go through, I mean, because it's really, pembrolizumab is really only for patients who are overexpressing PD-L1, right, on their tumors?
That's right. So the combined positive score, or CPS, is what's used, and that was used in the approval of pembro in these tumors. And CPS has to be 1 or greater. And the challenge is that there's a good number of patients. I don't know exact percentages, but probably on the order of 25 to 45% perhaps, that do not express PD-L1 and have a CPS of less than 1. And so for those patients, immunotherapy is not an option in the current state of affairs.
And I just want to bring up the role that HPV and HPV vaccination plays in the diagnosis and incidence of cervical cancer. Kind of maybe touch a little bit about on that.
Yeah, so the majority, and in fact, probably nearly all cervical cancers are the results of HPV infection. I think that, the HPV vaccine against the most common high-risk subtypes of HPV, 16 and 18 specifically, have shown real promise and are already decreasing some of the pre-cancer rates. I think the uptake of the HPV vaccine is clearly not what it should be. And my personal hope is that eventually I never see a patient with advanced cervix cancer in my clinic. When I was in medical school, I learned that cervical cancer was a disease of the past because we now had pap smears that detected cervical cancer. And so we could diagnose women early. And the reality of that is not all women get pap smears. Not all women have access to care. So there's a lot of disparities in cervix cancer. And I think it's going to also play a role in the uptake of HPV vaccine. So we've got some work to do on the prevention and screening front.
So the lesson is get your kids vaccinated.
That is the lesson. Mine are vaccinated, highly recommended.
Yeah,
people do not want to come see me in my clinic.
Yeah, these days with vaccine hesitancy on the rise, I wanted to sort of put the pitch in for vaccines. So at ESMO this year, like I said, there was a couple of drugs that were presented, and maybe we can talk about them, and we can kind of maybe compare the results and sort of see how they are similar or different. I don't know if you want to just kind of maybe go through what we saw at ESMO this year, and I can chime in.
Yeah, so I'm going to have to pull it up on my screens just so I can review those numbers. So there's 2 drugs that we specifically sort of chatted about in advance of this meeting. One was an ADC therapy, or antibody drug conjugate therapy. And I'm going to butcher the name because that's what I do with these novel drug names. I really wish there were simpler names. Tisotumab vedotin, or TV. And then another.
Call it TV.
TV. We're gonna go with that. Then I think that's perfect. And the other one I'm gonna butcher too, balstilimab, which we're gonna call BAL for the purposes of this conversation. And that's an immune checkpoint inhibitor. It's an anti-PD-1, so it is in the same class of drugs that pembrolizumab is in, for example.
So I think what was interesting about the BAL data is that they had responses, and I think their overall response rate, and I need to also look on my notes here, and correct me if I'm wrong, right?
Yeah, I just have it pulled up. So,
It was
BAL,
like 13-14% overall response rate. So similar to Keytruda or pembrolizumab in that, but they had responses in the PD-L1 negative patients, which is something noteworthy, something that we don't see with pembrolizumab. How significant is that?
well, I think it is significant. Again, it's too early to tell because these are, it's early data. One of the things that I did like to see about that study is that there were more patients in that study than in the the Keytruda study, the Keynote 158. So we do have a bit more data on these cervical cancer patients. But really for that patient population who does not have PD-L1 expressed, there's not an immunotherapy option. And so this would, if it makes it to the market, would certainly give those patients an option.
And to clarify the kind of patients that we're talking about in these studies, these are patients who have tumors that are not responsive to prior therapies, correct?
Yeah, these are women who have had prior treatment. Typically, they've had the prior chemotherapy with a platinum and taxane, maybe with or without bevacizumab if they were eligible. And then they've had progression or recurrence either during or after that treatment. And what we know about cervix cancer patients when they have metastatic and recurrent disease is that their likelihood of response to a treatment, a chemotherapy, traditional chemotherapy is very low. And for women who do have a response, it's very transient. So the current standard of second line chemotherapy is just not a standard. It's not good enough. So my recommendation to my patients is at progression, clinical trial is really the best option if they do not have the option of immunotherapy or if they have progression after immunotherapy.
Mm-hmm. Okay. And so again, you thought that the fact that they showed responses in the PD-L1 negative patients, that something was a differentiator from what we've seen with pembro.
Yes. Definitely.
Okay.
We don't have that information in the Keytruda study that I think it's indicated for those patients that are PD-L1 positive because that's where the activity was. And this is definitely a differentiator.
Right. Now with TV, tisotumab, a drug, like you said, an antibody drug conjugate. So talking a different class of drug here from Seattle Genetics, 24% overall response rate in their study, 7% complete response rate. Your thoughts about those data?
Well, I think it's intriguing. In that study, they also had prior chemotherapy and prior bevacizumab, the patients who were enrolled. I don't think they were allowed to have prior immunotherapy. So again, that's a population of patients that we do need to evaluate, and that wasn't studied there yet. But that response rate is much better than we would see with traditional second-line chemotherapy. So I would love to see more mature and larger data on survival outcomes. But in terms of response rate, I think it is intriguing.
Mm-hmm. So both the companies, balstilimab is Agenus, Agenus, and like I said, Seattle Genetics is developing TV. Both companies have indicated that they would seek accelerated approval based on these, so basically, based on as lots of cancer companies do, they go to the FDA with response rate data in these kind of heavily pretreated patient populations seeking accelerated approval. Your thoughts about, I know people get uncomfortable making sort of predictions about FDA, but like, what are your thoughts about these two from that standpoint?
Well, I think if you compared to the approval for Keytruda, there is a precedent for approving these drugs based on response rate. I've seen drugs approved and get pulled, once larger data sets come out. But certainly, I think there is a precedent for approval in drugs like this with these response rates.
Mm-hmm. And getting back to you, David, because I don't want to leave you out. I'll ask you the same question because obviously looking forward with AMG 510, Amgen is conducting a phase 2 study in about 100 lung cancer patients which would form the basis of a regulatory filing if positive. I mean, what your thoughts on kind of the data that you've presented and that sort of what we've seen, the profile, the efficacy, the safety profile, if that data were replicated in the phase 2 study, approvable? And then I guess maybe the question is like, how much lower could those response rates, maybe the durability of that drug go before you start to question whether or not it should be approved?
Yeah. These are really good, conjecture questions to sit around with your VC buddies. And I will tell you, going back to what MJ was saying about approval in cervical. By the way, I just want to shout out for tizotumab vedotin. I was the one who did the phase 1 and did the expansion in that, and we saw that cervical, and my colleague Rob Coleman, and
yeah, and you know how to say the name too. I'm impressed. Yeah,
and it's like each of these drugs are like children of mine. It's good to see that they're possibly going to get a job. But anyways, going back to what she mentioned about what the FDA prior, I think it's like the Supreme Court. Like if there's precedent, they can't go back against precedent, right? So I think that if it makes sense that Seattle Genetics is putting that to approval now. Going back to the phase 2, so the phase 2 in sotorasib, there's going to be 126 patients, and the question is will the response rate, will the progression-free survival be maintained in a similar, third line and above population? I don't know. I think so. That's my best guess.
Mm-hmm.
You saw, it's so you always see low number initial phase 1 data. We had, I think, I forget, 20 some patients, and then the next time we reported along, and there was a drop in the response rate. But 40 to 59, there wasn't really a drop in the response rate, and the progression-free survival was about the same. I don't know what the exact number is, but my best guess, and this is a guess,
mm-hmm.
for any regulatory people out there who are gonna tell me that this is my own guess. This is not Angie's guess, or my guess is that it's probably going to be around the same. We'll see. I mean, we'll see what the data shows. But
I mean, one quirk in the PFS curve on the data that you presented, was kind of and it's hard to describe without seeing it, but the PFS curve kind of flatlines right at median.
There wasn't any other events. That's just the nature.
Right. So it kind of looked like it could have been between 4 months to 6 months. It's.
Oh no! See, the 95 confidence interval rate is actually 4.8 to 7.9 months actually.
Okay.
So you could theoretically have a higher progression-free survival.
Mm-hmm.
Ultimately, I think all of these things like response rates, progression-free survival, they're surrogate markers to what we all hope for, which is overall survival, right? And there was a paper, by the way, by Blumenthal and Pazdur in 2015 looking at this in something like 12,000 patients in these randomized trials that the FDA had on patient-level data. And what they showed in that paper, this was in JCO 2015, Blumenthal et al., is that essentially response rates correlate with progression-free survival, and progression-free survival correlated with overall survival. So if they maintain this, if this overall survival and progression-free survival is around 6, is around 30, my guess is that the FDA, I mean, they're the ones who published that paper, right? The FDA is going to say, oh yeah, this makes sense, right? And again, precedent, they're like the Supreme Court. Precedent takes precedence, and they will look at that, and they've had other agents approved based upon overall survival and progression-free survival in non-small cell lung cancer. So I think that just speaks for itself.
Okay. MJ, question for you, and this is actually coming from one of our viewers. Wanted to know, assuming if both of those drugs that we've discussed become available, would prior bevacizumab influence your decision whether you would use an immunotherapy or the the ADC in second line?
I don't think so. I mean, I think the reality is if a patient can get Bev, they should get it, because that does give a survival advantage. And so once they progress, even after receiving Bev, I wouldn't hesitate to give either one. I think that the TV does have some bleeding complications listed in some of the side effect profile, but so does Bev. So if they were able to receive Bev, then I wouldn't hesitate. Now, if they did have a contraindication, such as to Bev, that might influence a choice of using TV.
Okay. And David, one of our viewers wants to know the JCO citation of the study you just mentioned, if you had it up the top of your head.
Blumenthal et al. I think it was May 20th, 2015. It was I would just put in Blumenthal and Pazdur, and I'm sure you'll get the paper in Google.
MD safety, we didn't really touch about. We talked a little bit about, but someone asked about the toxicity in both treatments for cervical cancer and risk benefit. Any thoughts on what you've seen so far from those data?
Well, the BAL data is very similar to pembrolizumab data. It's immune toxicities are really the what we see. It's not very different. I think with TV, it is a little different in that there are some ocular events, including ocular bleeding, which is not something that we want to have happen. And then peripheral neuropathy is a potential side effect of TV. And the challenge there is women will have had prior platinum drugs and probably prior taxanes as well. And both of those have neuropathy as a potential side effect. And so I do have women with cervical cancer, for example, who have pre-existing neuropathy that may limit the choice of options next.
Mm.
Can I make a comment on that
Yeah.
toxicity? So the ocular toxicity is real, but I don't recall we ever saw any patients with ocular bleeding. But there was definitely, it's tissue factor, so it targets tissue factor, right? So theoretically, you could have increased risk. Most of the bleeding events were really kind of minor nosebleeds. The ocular toxicity was real, but we've been able to, at least in we're in the phase 1 expansion, use steroid eye drops and cold packs. And that almost eliminated, I don't want to say eliminated totally, but it totally significantly reduced ocular toxicity. So, I think there's ways to kind of obviate those toxicities. It's not, it wasn't unmanageable, I guess.
And I think, David, I was misreading the abstract. You're right. It's bleeding events and ocular toxicity.
Yeah. So MJ, one thing we didn't talk about was the cell therapy that's also being developed in cervical cancer. These data from Iovance, they weren't presented at ESMO this year, but they've been out there previously presented. I think they presented some data at ASCO last year. And really high response rate from the cell therapy of 44%. In this similar patient population. Thoughts on kind of where that might fit in?
Well, I think it really is a promising therapy. I think, similar to CAR T and heme malignancies, TIL or cellular therapy in solid tumors is going to have a role. I'm not quite clear in my own mind on how that will actually fit in. I have spoken to Iovance actually about bringing that technology to my institution, and some of the hurdles that I see are that it does take a specialized center to perform that therapy. And so, with cervical cancer, there are so many disparities that exist as is already. Patients often have a hard time with travel and maybe underinsured or uninsured. And so outside of the clinical trial, there must be a way to have this ultimately, if it's approved, as an affordable, reimbursable at a decent rate drug that is accessible to those who need it. And that's really my concern with that therapy, but it does look quite promising in terms of those response rates. And it's a novel therapy. I think we need more of these. I just would love to see in parallel some thought to how do we not only come up with this great therapy, but how do we come up with it and then make it available to all patients who need it, not just patients who are commercially insured and live in an urban area.
And to clarify for any viewers who are unfamiliar with that, this is a personalized cell therapy. it's made for each patient.
Do you know if you
That's,
guys, is that a TIL therapy or was that?
it's a TIL therapy.
Okay.
Yeah.
Yeah, tumor infiltrating lymphocytes for the listeners.
These trials are really complicated to do.
So for the reasons, MJ, for the reasons that you stated, you would think, all be that would sort of be behind the, an ADC or another effective checkpoint in this disease.
I think it would, only because it's just not as accessible. I mean, this holds true for patients who need CAR T therapy for, relapsed or recurrent lymphoma and leukemia. And it's just not always feasible for all patients. If a patient doesn't have commercial insurance, institution may not actually be able to treat them because they lose money. And we don't need that added financial toxicity playing a role in whether we can treat our patients.
David, back to you. A viewer wants to know about any updates on AMG 510 in colorectal cancer. I know that the Nichen paper, I don't think there was a lot of new data in the Nichen paper when it comes to colorectal. Can you, any update?
There was a 7% response rate, right?
Mm.
So some patients respond. We don't know why yet. I suspect, and I think there's a lot of emerging data. There's a recent cancer discovery paper looking at EGFR overexpression, and there are investigators like Dr. Kopetz here in our group who really believe that kind of this adaptive response through the EGFR pathway is probably and likely the mechanism of resistance to 510 in these G12C patients. So, there are ongoing combos, both Mirati and Amgen will test this hypothesis. I personally think that there's probably going to be, additional benefit with standard chemo like irinotecan and so forth, but we'll see how that plays out. And, but I think the story here, and again, I have to couch this because we don't know yet. I mean, I may be wrong, but the story is a lot like the BRAF story, right? So the BRAF story, you saw 60% response rates in melanoma, and then you saw like 5% response rate in colorectal. And if you talk with Keith Flaherty, who was the developed sorafenib, he would have told you that before he got this trial in phase 1 that everything was going to work in colorectal. And by Keith's brilliant, but to a large extent he became a melanoma doc because sorafenib had such incredible activity in melanoma relative to colorectal. And then later on, Prahlad et al. Published in 2011, I think, a Nature paper that EGFR may be a mechanism of adaptive resistance. We tested this combination of vemurafenib, irinotecan, and cetuximab in the clinic that led to an NCCN approval and positive phase 2 SWOG, and that led to the Beacon trial, which you all are familiar with, which also led to recently encorafenib and cetuximab being approved. So I think the story is going to be a lot like that, but we'll know, we'll see.
And that story, the combo story for KRAS inhibitors, is a story for next year. I mean, the combination studies are underway.
There's a lot of combos, and I think there's going to be lots of combos for years to come, to be honest with you.
Is there a combo that you're aware of that you're most excited about?
Well, I mean, the EGFR in colorectal is exciting, but I think a lot of these are exciting. I mean, I'm very excited about SHP2 and how it interacts with KRAS, but it also has an immune component on the tumor microenvironment. Obviously, we're all anticipating that the Pembro the PD-1 plus 510 combinations and also MRTX combinations will play out. Hopefully, those will be, we'll find a therapeutic window which will see synergistic activity. All these things, there's a lot of exciting things to come out in the future.
Well, while you have your predictor hat on, any thoughts about the Mirati? That's it's the other KRAS G12C inhibitor in clinical trials right now. They're going to have data in October, I think, the triple meeting. Any thoughts about what we'll see there?
I think it's very similar to 510, right? And you couldn't have predicted that necessarily with all the RAS inhibitors, right? I mean, that's why Lilly and J&J are no longer in the game, right? And now Roche is putting their molecule in, and there will be others, that have something in the background that will eventually put their hat in the game. But I think Mirati, at least, their data was much more preliminary, looks very similar to 510. I think they got a little bit lucky on the colorectal. They had 1 out of 4 responders in colorectal, so it looks like it's a 25% response rate. But I think that if you increase the numbers, you'll probably get down to around the same number with 510, but we'll see.
We are out of time. I told you guys in the prep that the time like flies by, and it has. Any last words, parting advice, any thoughts you guys want to share about anything that we've discussed or haven't discussed?
It's just an exciting time for oncology drug development, let's say, and many hopes for our patients.
Yeah, I completely agree. And I'll just remind folks, vaccines are good, and wear a mask.
There you go. And vote.
And vote.
Yeah.
Well, look, David, MJ, I really appreciate your time today and offering your thoughts on this topic. Lots of interesting things, and we'd love to have you back at a future event. We'll see how things go in all the drugs that we're talking about. And I want to thank all of our viewers for participating and hanging in with us this afternoon. And thanks again to our sponsor, Natera, for sponsoring this event. As you guys know, we do a lot of virtual events these days because we have to, because that's the way events, so we do a lot of these Zoom calls. We've got a full schedule of events here at STAT, and so stay tuned and check out our website for our future events. And with that, we'll end it here. MJ, David, thank you so much for your time.
Bye.
Thank you.